Well, I'm going to introduce our next presenter. Dr. Ehrlich, Dr. Garth Ehrlich, is a professor of microbiology and immunology and a head and neck surgery at Drexel University College of Medicine. He also directs both the Center for Genomic Sciences and the Center for Advanced Microbial Processing. It's hard for me to introduce all these scientists. I'm a lay person here. Within the Institute for Molecular Medicine and Infectious Disease and the Core Genomics facility within the Clinical and Translational Research Institute. So you can finish reading his bio in your program, but please welcome Dr. Ehrlich.
Thanks, Bobby, and thanks, Tammy and Jessica. So I want to add my welcome to everybody that's here. I'm going to try to teach you a little bit about how bacteria cause disease. And so I'll talk somewhat in parables because my lab has been studying different types of bacterial infections for nearly 30 years. And what I want you to understand is that what the Lyme agent does, although it's pernicious, is not that unusual. There's other examples. They just haven't been politicized, as you heard about from Mary Beth earlier. And so I'm fairly new myself to the Lyme world, but having worked in bacterial pathogenesis or how bacteria cause disease, as soon as I came into the field, I could see lots of parallels. And now, just like I did yesterday, I'm going to devote this talk to Bill Costerton, who was kind of a mid-career mentor for me. And if any of you have heard of the term biofilm, he was the guy who first coined the term. He's probably the most important microbiologist of the last 50 years. And so much of what I'll be talking about today is work that Bill and I did together over the last 20 years or so. So there are a couple dichotomies or contrasts that I want to convey to everybody. So probably everybody now has heard about chronic infections and acute infections, okay? And many types of infections have both chronic and acute forms. So acute forms are ones that come on quickly. You're usually very sick for a short period of time, but it resolves and you get better. Chronic infections tend to come on more slowly, but they're much more persistent and very difficult usually to get rid of. And so what we've learned is that there is a difference in the way the bacteria grow in general if you compare a chronic infection versus acute infection. So acute infections are what physicians and microbiologists have studied, for the last 100 or so years, really since the dawning of age of modern medicine. And so acute infections are generally associated with the bacteria growing as single-celled organisms, how we usually study them in the test tube, so in individual bacterial cells. Whereas chronic infections are generally associated with bacteria taking this biofilm form. And I'm going to talk a lot more about that as we go ahead. But just for now, remember that this is a multicellular form of bacteria. It's truly multicellular. If other people say, well, this is an agglomeration of bacteria stuck together. It's not. There are some other really pretty common and profound differences between acute and chronic infections. Acute infections are generally caused by a single strain of a single species. Not always, but usually. And chronic infections are often caused by multiple strains of the same species. And they can even be caused by multiple infections with different species. So, of course, you're probably very aware of that because so many of these tick-borne infections are polymicrobial. Somebody's got babesiosis as well as Lyme disease as well as anaplasmosis. So why is it important to know that you have more than one strain or more than one species present during an infection? Well, it's because it provides, and this is some lingo for you, it provides the genetic fodder, if you will, to the infecting organisms to evolve during the infectious process. Bacteria don't have sex like we do, but they do exchange little bits of DNA. And it's called horizontal gene transfer. So that's their equivalent of sex. And so if you have multiple strains or multiple species that are fairly closely related, they can constantly create new strains during the course of infection. So your immune system is constantly chasing a moving target. So the biofilm paradigm came about because in the early 90s, I was approached by a young ENT surgeon. That's why I'm a professor of otolaryngology. I'm not actually a surgeon, but I run the research program for the department. Because he came to me and he said, well, can we use PCR, that's the common DNA diagnostic, okay, to look for bacterial presence in children who have chronic middle ear disease? And for those of you who may or may not be familiar with middle ear disease, or we call it otitis media, is the most common reason, that children see a healthcare provider. It's the most common reason that antibiotics are prescribed for children. And it's the most common reason for surgery. They put tubes in kids' ears when they have the chronic form of it. So everybody knew when Chris first came to see me that acute middle ear disease was a bacterial infection because you could treat it with antibiotics and they got better. And you could also go through the eardrum and pull out a little bit of fluid, and you could stick that fluid in a test tube and the bacteria would grow. So nobody questioned that this acute form of this disease was infectious. However, by the early 90s when I first started working with Chris, the dogma had grown up, does this sound familiar? That chronic middle ear disease was not actually an active infectious process. It was resulting because of the body's response to some previously cleared infection. And the reason that people thought, you could understand why, because you would treat these children with the chronic form of this disease with high doses of antibiotics over and over again, and it didn't resolve. They'd still have fluid in their ears. And so if there was an infection, it should go away, because that was the paradigm that we had, for antibiotics and bacteria. And the other thing is you could tap their ears just like you would the acute kids, and you take some fluid and put it in the test tube, and nothing would grow in the vast majority of cases. So they had reasons why they thought that way. I got these slides out of order. So what I'm going to kind of do in reverse order is, what is a biofilm? So remember I said at the start that a biofilm is a multicellular organism. So it's not just a bunch of bacteria stuck together. If the way to think of it is that everybody knows about how if a caterpillar, goes through metamorphosis and it turns into a moth or a butterfly. The change from bacteria going from single-celled planktonic bacteria to a biofilm is more profound metamorphosis than a caterpillar going into a moth. Because there you've got a multicellular organism changing into another multicellular organism. Here you're going from a single-celled organism going to a multi-celled organism. And this was really a very profound change, and this was what Bill Costerton recognized probably about 40 years ago now. And so when we got involved in this, when we were studying middle ear disease, I was reading some of Bill's work, and I thought, oh, maybe biofilms explain what's going on with these kids with chronic ear infections. And so that's how, this was about 25 years ago. So we kind of brought that paradigm, because Bill was not primarily working in infectious disease. He was what we call a microbial ecologist. So he's saying how bacteria grow like in streams and in hot springs and that kind of thing. What we did is we brought his paradigm to medicine. Now, there were a couple other groups that did it at the same time. We weren't the only ones. And so the other thing that we realize now is, even though we've been studying planktonic bacteria, the single-celled bacteria, for really a very long period of time, that the biofilm is probably actually the ancient form that evolved first. We have fossil records of what are called stromatolites, these fossils off the coast of Australia, which are essentially fossilized biofilms that are 3.3 billion years old. And so we really think the planktonic form evolved, if you will, later as a way to disseminate and seed new areas. So the biofilm is actually very ancient. And we've not found any bacteria, with the exception of a few intracellular parasites, that can't form biofilms. So this is universal across all bacteria, whether you're talking about the ones that cause middle ear disease or whether you're talking about Borrelia burgdorferi, which causes Lyme disease. This is just the sine qua non of bacteria. So just to kind of review some biofilm concepts, they are the predominant way that bacteria grow in both natural and in infected ecosystems. Pure culture planktonic growth is rarely how bacteria exist in nature. And so as a pathogenic mechanism, or how something causes disease, it's really what we call a population level virulence factor. So most people, when they think about virulence factors or things that cause disease by bacteria, you think about something like a toxin which makes you acutely ill. When bacteria form biofilms, what they're trying to do is persist. And so their pathogenesis is related to persistence, not towards making you acutely ill like what you do with like a toxin or something like that. So I kind of went over this. So this was just some of the work that we did to show that indeed our hypothesis was correct. So the first thing that we did is we used an animal model. You've heard Monica talk about her models, using the monkeys. Well, we used actually the chinchilla as a model for middle ear disease. They're small animals, but they have very large ears and middle ear spaces, and believe it or not, they're actually very closely related to humans. We found this out. We did a chinchilla genome project a few years ago. And so what this was is we took the chinchillas and we infected their middle ears with a bacterium called Haemophilus influenza, which is the same organism that causes many of the middle ear infections in children. And what you're seeing on this slide is called a live dead stain. And so if the bacteria are alive, okay, it will stain them green. If they're dead, it will stain them red. It will also stain the host cells red. So the big red blobs that you see over there, those are the host cells. Those aren't bacteria. That's actually the mucosal layer, if you will, from the chinchilla. These chinchillas were treated with multiple rounds of antibiotics, okay? And yet there is no death among the bacteria. They're all bright green. They're all very, very happy. So this is the thing that you have to understand. Antibiotics, and you've heard this before, but it's worth repeating. Okay? Antibiotics are essentially not effective against bacteria growing in a biofilm. Okay? Our antibiotics were designed to kill rapidly dividing and rapidly metabolizing bacteria, because in the end they all work through the same mechanisms. And so then we actually then took it to the human model, and working with our colleague Joe Kirshner at the Medical College of Wisconsin, he actually was able to get biopsies from children that he was putting tubes in their ears, and he sent them to us. And you can see essentially the same thing. The red, again, is the host cell. These big ones here, these are like white blood cells. But these huge green concretions here, these are pneumococcal or Streptococcus pneumoniae biofilms. This was a patient who had failed 9 courses of antibiotics. And there are no dead bacteria here. So this biofilm paradigm, which we developed to study middle ear disease, actually turns out it really kind of works for all chronic bacterial diseases. So we did this original work in the mid-late 90s on into the early aughts. And so it turns out that almost all the chronic diseases that ENT surgeons see, in addition to otitis media, with adenoiditis, you get your adenoids removed, or tonsillitis or sinusitis, these are all biofilm diseases. And it's across all medical and dental specialties. So like if you're looking at like urology or gynecology, prostatitis, endometritis, cystitis, vaginitis, these are all biofilm diseases. In orthopedics, osteomyelitis, bone infections, if you get an infection of an artificial joint, these are intractable biofilm infections. In dentistry, you've got periodontitis, apical periodontitis, or what most people call a root canal. These are biofilm diseases. Zoonotic diseases, Lyme, brucellosis, Bartonella, they all form biofilms. So what I want you to understand is that Lyme has certain unique aspects to it, but it fits within a much larger model. And it's just that for some reason the rest of the world has, although it takes a few years, has embraced that these other chronic diseases are actually infectious. We had to go through each one. We first started with the orthopedic surgeons, and they said, oh no, these are aseptic loosenings. And then we showed, well, no, there are actually bacteria here. So, but the difference is the communities in these cases accepted, it took a few years, but they accepted what we were saying. We'll eventually get there with Lyme. It's just going to take a little bit longer. So this is not my work. This is actually one of our colleagues who works, Ava Sappi. This is just an image from one of her papers showing that Borrelia burgdorferi also forms biofilms. So it's no different from any other bacterium. And so one of the things, I hope this will work, that I want people to understand is that biofilms often form at distant sites in the body from where infections are. So if you have I was talking about if you get an artificial joint that gets infected, okay, the infection doesn't actually start there. It'll generally start because perhaps you had a dental procedure, you brushed your teeth, and then some of the bacteria that were on your teeth to get into your bloodstream. And then they pass through your bloodstream and they find a suitable site. They'll land on this big piece of metal, which is your artificial joint or something like that. And then they'll form a biofilm there. So my colleague, Mark Shurtleff at the University of Maryland, prepared this. I'm going to try to see if I can get this to run. So this was drawn for Staph aureus, which is another bacteria probably most of you have heard of. But the same model works for any sort of bacterium. So it's broken through the skin here. Now it gets into the blood vessels because all these bacteria are what are called adhesins, so that they can dock to host endothelial cells and then pass through. So now you've got a little break in a blood vessel. So now the blood cells come out, but a few of the bacteria come out as well. Then they attach, and they start dividing, forming a biofilm. So this model of this was drawn for Staph aureus. Works essentially for any bacterium, that gets into the bloodstream or into the lymph nodes. It can pass through the body, and then wherever it finds a suitable place, it can form a distant, site of chronic infection. So we come back to why are biofilm bacteria recalcitrant to antibiotics? Well, when you have a biofilm, all the bacteria are not doing the same thing at the same time. And everybody knows from almost every walk of life that diversity provides strength. And biofilms are very heterogeneous. So you've got different bacteria are doing different things. And so even if the antibiotic does affect some of the bacteria in a biofilm, it's not going to get them all. The other thing is, that we now know, is that the bacteria, they're true multicellular organisms. And so when a biofilm gets hit on the edge with a noxious agent, like an antibiotic, it will actually send signals to the other bacteria or the other, that are deeper in the biofilm to upregulate their defenses against antibiotics. So when people say these organisms are smart, they're not kidding. They are really smart. Okay? And they have every type of mechanism that we have for communicating within our bodies, they have. They have primitive nervous systems. They have the equivalent of what we call cytokines. You may have heard they have cytokines. It's called quorum sensing molecules. They release these little membrane-bound vesicles, which are just like what our nervous systems do, and that's what traffics very rapidly through the biofilm. And they also have these nanowires so they can conduct electricity through the biofilms. So the more we study, the more that we realize that a bacterial biofilm is just like any other multicellular organism. And antibiotics, as I said, were developed and designed to kill rapidly metabolizing, dividing bacteria, planktonic bacteria. So this is just a picture to show you the heterogeneity within a biofilm. So what we're just doing, looking at the top, is like at the surface of the biofilm, and down at the bottom is the base of the biofilm. And it doesn't matter what the colors are representing. They're simply showing you that there's different metabolic conditions depending on where you are in the biofilm. So at the top, the green means they're rapidly metabolizing. In the middle, there's kind of a little bit of a yellow area, or they're kind of intermediately metabolizing, and there's a huge area below that, which is all red, where they're very slowly metabolizing. So how do antibiotics function? Well, we have many different classes of antibiotics, and each one attacks a different part of how a bacteria grows and divides. But in the end, they all kill by the same mechanism. You heard Dr. Stram talking before about reactive oxygen species and the damage that can do to humans. Well, reactive oxygen species will kill bacteria too. And in the end, all antibiotics kill by essentially forcing the bacteria to produce high levels of reactive oxygen species. And so that's actually what kills the bacteria, not the fact that you stop it from forming proteins or that you stop its DNA replication. It's because you threw a monkey wrench into this high-speed process, and now you've got all these mechanisms that are just spinning away and spitting out unquenchable reactive oxygen species. So it's oxidative stress. That's how antibiotics kill. Originally, it was thought that resistance was due to lack of penetration. You'll hear lots of people still say that. They'll go, well, the antibiotics can't penetrate into the biofilms. That's actually not true. Phil Stewart and I showed in the late 90s that the antibiotics do get in, they can penetrate. So that's not the issue. I felt it was due to what we call this metabolic quiescence. And so we showed that by limiting oxygen, that it would provide protection, because if you don't have oxygen, you can't make reactive oxygen species. And we showed then, using oxygen sensing microelectrodes, that the cells deep in the biofilm, they're actually not getting any oxygen. It's actually what we call anaerobic. There's no oxygen getting, because the guys up at the surface, they're greedy. They suck up all the oxygen before it even can penetrate down deep. So it's the guys in the bottom that are what people refer to as the persisters. Okay. They're in this oxygen-free zone. And we showed then that if we gave the bacteria something that they could ferment, okay, a different way of growing, then we could actually activate their metabolism and then start killing them with antibiotics again. So that's how we knew that it was metabolic. And this is just an image of just what happens to oxygen concentration as you go through a biofilm. And so you can this black bar just saw how steeply the oxygen concentration dropped. So this is oxygen concentration on this axis. And what you see is that by the time you're just a few microns into the biofilm, the oxygen has dropped, to almost nothing. I'm going to skip over this. So after we'd done all this work, we thought that the reason that they were resistant to antibiotics was because they were passively resistant, if you will. It was just because they were metabolically quiescent. We were wrong. And I was so embarrassed when our colleagues published a paper, Pradeep Singh's group. I was actually the one who was the reviewer of the paper because I've been working in the area, and I'm thinking, oh my God, I could have had a V8. But basically what he showed was that the persistence or the resistance was part of an active process that the bacteria trigger when they're in an oxygen-limiting environment. And it's called the stringent response. And the stringent response is what turns the bacteria into a persister cell. And what you need to know is that a persister cell bacteria is really the equivalent of a stem cell in a human being. And what's the difference between all the other cells in our body and a stem cell? Stem cells can live for decades, more than a century. If you've got somebody who's 100 years old, they've still got stem cells. Those stem cells have been there since birth, okay? The difference between a stem cell or a persister cell is it's not trying to divide. It's trying to survive. And so what it does is it massively regulates, get this, its ability to deal with oxidative stress. Okay? That's why the antibiotics don't work, because the persister cells have elevated the enzymes that allow them to deal with oxidative stress. You heard from Dr. Stram, they're the same in the bacteria as they are in humans. Glutathione, superoxide dismutases. So this stringent response is what's called a general alarm system. And it's very ancient. part of protein synthesis, if you will, which is the most conserved mechanism across all bacteria and all life. And so once we realized this, we realized that we had a molecular target where we could develop a drug against the molecule that triggers the stringent response. And then, so the idea would be then, if you can stop the trigger of the stringent response, then you can repotentiate antibiotics against biofilm bacteria. Because we already showed, and Pradeep's group showed, that if we take a bacteria that we've genetic engineered and we've removed this enzyme is called REL-A, that they'll form biofilms. They look a little bit different. They'll form biofilms. But they die with antibiotics just like planktonic bacteria. So I kind of went over this already. So this is just what my medical colleagues call a Ph.D. slide. It shows the pathways that of metabolism that REL-A affects. And this was the molecular structure of the REL-A enzyme, which was just produced about a year and a half ago, but it allowed us then to use structural methods to actually go in and identify what the active site is. That is the part of the enzyme that actually produces what we call this alarmome. Okay, it's called magic spot. And so then what we did is we tried to find molecules that would fit in that same spot. So this region here, this is the active site of the enzyme. This is the business part of the enzyme right here. And so we were able to find molecules by computer that would fit very well in that site. And so this is one of these green here is this one compound that we found doing what's called in silico testing, which is the way everybody starts out doing drug discovery now, or usually, because you can screen millions of compounds in the computer before you actually start working on individual ones. So you get your initial ideas from these computer screens, and then you actually go in the laboratory and see if they do what you think they're going to do. And so I'm probably running very short on time. I'm just going to kind of skip ahead and say that these compounds work as we predicted in the laboratory. We haven't gotten into animals yet, okay? But we can show that they work synergistically with antibiotics to kill biofilm bacteria. And that's, and so now we need to take these, into the animals, show that they don't kill the animals, they're not toxic to the animals, and that they actually will break up, biofilms in the animals. And we're hopeful, as I was saying, that they may actually be more effective in an animal than they are in the test tube. Because if you disrupt the REL-A, as I said, it still forms biofilms. But these are biofilms over here which have not been treated with our compounds. So you can see they're just these kind of massive, flat, dense glassy surfaces. And these are biofilms here that have been treated with our compounds. So it's a much more open structure. And one of the problems that our white blood cells have, neutrophils, usually that are supposed to try to help mop up biofilms, is they can't penetrate into the biofilm. So we're hoping that if you treat with antibiotics and this drug and the fact that it's got this more open structure, that it may be a little easier for the white blood cells to come in and mop up. Again, this is just wild speculation on our point, but we're hopeful. So thank you.